Good evening, everybody. Sorry about the slight delay, which was caused by me just losing my internet connection at the crucial moment. Classic piece of 2020/21 lifestyle, which is very appropriate for this evening because our theme today is a journal of the pandemic year. And with us this evening, we have Professor Richard Cornall, the Nuffield Professor of Medicine and a Fellow of Magdalen College. Richard was educated first at Trinity, Cambridge, then at Balliol, and then at London, before becoming a research fellow in immunology at Stanford, and then returning to Oxford as a honorary consultant in renal medicine and professor of immunology and a fellow at Corpus Christi. In 2019, he was appointed Nuffield Professor of Medicine and a Fellow of Magdalen, succeeding Sir Peter Ratcliffe in that position. Richard is the head of the Nuffield Department of Medicine at Oxford University, the largest department of medicine by research in Europe. And he is therefore particularly well placed to talk to us tonight on the last year of the pandemic. So without more ado, I'm going to hand over to Richard. If you have questions, the usual format applies during these webinars. Ask any questions in writing in the Q&A box. I will put those questions to Richard at the end of his talk. So I'm now going to hand over to Richard, and thank you very much.
Okay, thanks, Diana. I'm going to share my screen. And I hope you can see my slides now. Okay. So thanks very much, Dinah. So for any of you who haven't read it, I'd like to recommend the Daniel Defoe's Journal of the Plague Year. I mean, it's a brilliant description of what happened back in 1665, full of complacency about the disease, fake news, panic buying, lockdown, unequal distribution of resources, and so on. And this quote that I gave you was kind of very redolent of I think at least the first lockdown. And the last time I was in dinner in college, I was sitting next to Marcello Catania, who's one of the fellows by examination. He's working on this period, and he was telling me about Daniel Defoe's excellent storytelling. And it's curiously accurate when you see that this was written when Defoe was 5 years old. But I think it shows that these sort of cataclysmic sort of events, last long in our consciousness, and it's undoubtedly true that this last year is going to be the same. And so this is going to be my sort of abbreviated diary of the last 12 months. But before I get going, I thought I'd give you a little bit of background about the pandemic. So you'll be aware that this is not the last plague that's affected humankind, and we know that the Black Death in the 14th century killed roughly a third of the population of Europe, and obviously the same infection in 1665. It was caused by a bacterium, Pasteurella pestis. That's now quite treatable with penicillin. Most of the pandemics, though, have been due to viruses, and the last major pandemic was the influenza pandemic in 1918, which killed more people than the Second World War. And we continue to see threats. In the last really worrying threat was the Ebola crisis of 2014-16. That there were 28,000 cases in West Africa, and the mortality was 43%. If you go back in history, there are lots of other cases, less well defined. The Antonine Plague in the 2nd century killed Lucius Verus, who was out fighting in the Eastern Empire, and then spread to killed probably a quarter of the Roman Empire at the time, including probably Marcus Aurelius. But these events don't predate early history. So we've probably only been having these pandemic events since around about 5000 BC, because they need a certain density of population to maintain them. So when we were living as hunter-gatherers in dispersed populations, we weren't significantly affected by these infections. We just weren't simply able to sustain them in the population. So we know that the Antonine plague, which was described by Galen, was probably smallpox. So if you look at the sequence of smallpox, you can deduce that it arose probably about 3000 years ago, and it jumped from a rat. So African rat pox jumped to smallpox, and its mortality was around about 40–50% when it was eradicated in the 1970s, and it's similar to measles. Measles, less severe, but also jumped from cattle. Rinderpest. Rinderpest, like smallpox, has been eradicated by vaccination. Measles should be eradicated by vaccination because although it mutates like all viruses, its receptor that binds to our cells is very intolerant of mutations, and really this ought to be eradicated. It's less clear when measles arose. Now, of course, if viruses like these can jump from animals, then coronaviruses must have come from somewhere too, and there are two coronaviruses which cause common colds, and it's hypothesized that one of these infections, which is this OC43, may have been the cause of the Russian flu pandemic in 1890–92. It's not certain, but and perhaps it's more likely, in fact, that it's flu. But this hypothesis is very interesting. That the Russian flu killed 1 million people out of a population of about 1.5 billion, including Prince Eddie, who was the elder brother, less adequate elder brother of George V. And the importance of this, I think, conceptually, is that if something like a coronavirus causing the common cold could cause this number of deaths, it might be that it infects the human population. It causes a large number of deaths, like the current pandemic, in older people, but then becomes sustained in the population because it doesn't affect younger people. We develop immunity, and then it perpetuates itself in a milder form. And this may well be how the current pandemic is going to evolve, which I think is something to in a way to take some heart from. Okay, now, so coronaviruses themselves, we've been affected by coronaviruses in recently and in the recent past, and the last time was when we had SARS-1. That was an infection that arose in Guangdong province, just north of Hong Kong, originating in bats. It spread to nearby Hong Kong, and there were quite a number of cases in Canada. There was quite a lot of secrecy when this outbreak occurred amongst the Chinese, and so some of the first cases were recorded outside China in Vietnam. And Peter Horby we have a research station in Hanoi, and Peter Horby, one of our researchers, was running that, and he described some of the first cases of SARS-1 outside China. And SARS-1 is very similar to SARS-2. The spike on the virus binds to the same receptor in humans. The pattern of disease was very similar in that it affected older people, and the R number was similar, but it was different in that most people were symptomatic. And so there were over 8,000 cases, a large number on ICU, and several hundred died. But it was eliminated by track and trace. And excuse me. The importance of this is that it helped to shape our the thinking for it and diseases like Ebola and others, helped to shape the way in which we planned pandemic planning around the world. And there was a kind of assumption that this clinical severity of disease would be proportionate to numbers of seeking help. And so it was on this basis that we were preparing for pandemics in the UK and in other Western countries. And so the idea was, if we had SARS-1, there'd be a significant number of unwell people, but they'd all be obvious, and therefore we'd be able to track them down. And there'd be very small numbers of asymptomatics or mild cases. So conversely, if you had a disease like a pandemic caused by influenza, there might be many more mild or moderate cases, but the number of severe people, cases in hospital would be quite manageable. And then in 2009, the last declared pandemic before this one was an influenza pandemic, H1N1. We were very worried about this because we thought this might be like the 1918 pandemic, but the hospitalizations were actually 40 times lower than with the SARS-2. And it's kind of now a sort of seasonal flu that we manage with vaccination. What nobody had imagined was that we would or plan for at any rate, was that we would have a virus like the current SARS-2 virus, where we'd have 80% of the population would be asymptomatic and carrying it around, but there'd be a significant number of people with disease that we wouldn't be able to manage in hospitals. And the reason why we've struggled is partly because of this, these assumptions that we made, I think, about planning. Okay. So if you want to understand the disease in more detail, then you need to separate the virus, the transmission of the virus, from the effect on the host, which is the disease that it causes. And so just conceptually, to run through this, what happens in the early phase of the infection is the virus jumps from the host, which in this case would be a bat. It's covered with these spike proteins, which by chance also bind to our ACE2 receptor. It then evolves because it needs to bind, it can bind these more optimally, and because it mutates and there are billions of copies, it starts to optimize its transmission. And that's the state that we're basically in now, where these viruses of variants have formed in the Kent virus, which we noticed in the UK. And we primarily noticed in the UK because we were doing a lot of sequencing. And so we noticed this in the UK, and it became the dominant strain simply because it improved the transmission, because it bound the receptor more effectively. And so that's what happens in the first phase. And then our immune system responds to this as we become infected. The number of infected is not high enough for herd immunity at the moment. And so we're developing antibodies against the spike protein and other proteins in the virus. And some of these antibodies bind to the spike protein and prevent it from binding to the ACE2 receptors. And these are known as neutralizing antibodies. And the strategy behind most of the vaccines has been to try and immunize people with the spike protein and generate these neutralizing antibodies, and thereby prevent the transmission of the disease. Now, of course, the virus can also mutate the spike protein again to avoid the antibodies, but it's rather harder for it, and it's constrained because it still has to bind the ACE2 receptor. But this whole process is extremely dynamic and continually evolving. Now, in contrast slightly, the thing we're really worried about is the host disease. And this is, most people think about this as, we've got to fight off the infection, but actually it's both fighting off the infection and being tolerant to the infection that's important. And it's a bit of a Goldilocks situation. You don't want to produce too much immunity. And in fact, the reason why older people have been dying is really because they're not tolerant to the effects of the infection. And that's evidenced by the fact that the best treatments that we've developed over the last year have been mainly to dampen down the immune response. Now, this part of our immunity is largely governed by a different subset of cells called CD8 T cells, which are kind of killer cells that kill virally infected cells in our bodies. And they are generated again against these antigens in the virus, but against different parts. They're not the bit that binds the ACE2. That's not the critical bit. They can respond to any part of these viruses which are detected within cells. And it's important that the viruses the vaccines also produce these CD8 T cell responses, and we can measure them. Now, as time goes by, the host also evolves against the virus to become more tolerant to it, and that's a natural history of our response to these viruses that jump from pathogens. And this is a much more durable evolutionary response because it doesn't disadvantage the virus, so there's no competition, but it's very slow. But if you look in history at other viruses where we know they've jumped, for example, smallpox, and you look at the instance of smallpox in 1500, it was the mortality was around 30 to 40%. But if you look at what happened when there was migration to the American littoral with people from Europe, the mortality in the people they encountered was more than 70%, and that's because there must have been some evolution to create greater tolerance against infection within the European population over the sort of 2,500 years or so since it jumped from the African rats. And of course, these people were isolated because the Bering Strait migration was about 10,000 years ago, in hunter-gatherer time. The same thing applies to rats. So, to bats. That they have probably, the reason why they are probably the host of these coronaviruses is probably because they've been living in these sort of densely packed sort of urban-like structures with very high levels of migration for probably 100 times as long as humans have had city-states and have been living in close proximity. And the bats tolerate the coronaviruses because they miss a large part of their immune response, and so that's probably why they're hosts. Now, this separation of transmission and disease is really important for the vaccines, as I've kind of alluded to. And a good example of this is polio vaccines. So back in the 1950s, the first polio vaccine was produced. It was a killed polio vaccine, and it was injected, and it was really effective at preventing paralytic polio and infection of the nervous system. And it produced these sorts of immune responses that I've described on this side, but it didn't produce good enough neutralizing antibodies, and it didn't prevent transmission. And so we reverted to a different polio vaccine called the Sabin polio vaccine, which you take orally. It's the one on the sugar lump, and that produced high neutralizing titers and prevented transmission. So that's a really kind of nice example, I think, of how you've got one which presents disease, and you've got another that prevents transmission and probably disease as well. So this has arisen very recently in the press because everybody's worrying about the neutralizing antibody titers of the Oxford vaccine and other vaccines that are being produced worldwide. As the variants arise in the spike, we know that the neutralizing antibodies that we've generated against the previous versions are not quite so high. But the same antigens, same vaccines are producing really good T cell responses. And it's probably that's the reason why and the reason for us to have good confidence that that the vaccines are going to be nicely protected against disease going forward. Not yet proven, but there's starting to emerge evidence from people that have been vaccinated in Brazil with the Oxford vaccine, where they have a different strain looking like they're beginning to get protection. And we'll know that, we'll have that evidence in the next few weeks. So that will start to come out. When we have that, then we'll have a much greater surety that that will be ameliorating disease regardless of these variants and the neutralization. Of course, that means that the new viruses, new variants may spread, but they won't be causing significant disease. And we've got data just recently showing that the T cell responses to the spike from the Oxford vaccine are even with the variants, are really well maintained. So that's all very reassuring. Okay, so that's all the background I'm going to give you. And now I'm going to get on to the journal bit. So the start of the story for me is that when, so Peter Horby, who I told you about in Vietnam, he came back to Europe, to Oxford in about 10 years ago. And And one of the things he and Jeremy Farrer, who's the director of the Wellcome Trust now, did is they set up something called the International Severe Acute Respiratory and Emerging Infections Consortium. And it's a sort of grassroots organization for pandemic preparedness. And part of the rationale for this was that when new infections arise and pandemics, you don't have enough time to create the research infrastructure that you need in order to manage the pandemic as it breaks out. You don't have the trial protocols and all the infrastructure you need. And so the idea of ISARIC was to put all this stuff in place. And so when we had the outbreak in December 2019, all these protocols and structures were already put in place in China and were created really by this organization that grew out of our department. And so we were very aware of this from the very beginning. The first paper that was produced in China, published in January, used the ISARIC protocols. And so, and the first, and so this, another thing was that so we were very connected with everything that was going on in Wuhan, although it was locked down. The director of the Chinese, Center for Disease Control, this guy George Gao, he was a postdoc of John Bell's in Oxford. Before he went back to China. And so he led the response. And unlike the previous SARS episode, they were very open and there was a lot of communication. And this survey, that was published by Peter Horby and George and others, described the sort of early timeline. So you can see that here's the first case. Here on January the 1st, the seafood wholesale market was closed. This was the first fatal case, was on January the 11th. They published the sequence of the genome of the virus on the January the 12th. That was important because then everywhere in the world we could all start sequencing and making proteins, and then gradually it started to escape, and the first cases started being reported outside China. And it was about this time that Peter Horby came to me in my office and said, this is not going to go away, and we've got to start doing something. And so we called together a meeting, and we had an hour-long meeting on the 30th of January sorry, let me go back, in Oxford. And I called together a whole load of people from across the department and wider university, and we just sat down and thought about what we could do to help. And on this call were a few people that some of you will know. So this is Adrian Hill, who's a fellow here at Magdalen, and is a director of the Jenner Institute, and Sarah Gilbert was there, and other people some of you will have seen on the news, Theresa Lamb and Kath Green. This is Gavin Screeton, who's the head of the medical sciences. This is John Bell was there, and a lot of other people. And this is Miles Carroll. So just a month before we put this on, we had this meeting. I'd appointed Miles Carroll part-time to the department, and he's the director of research at Porton Down. So that was a very useful contact. And then we had our directors from our overseas units were also there. And so there were kind of two major decisions that we had at the department at that time. And these were, the first decision was that we decided that we'd start making high quality proteins of the virus. And this was partly motivated by the report from Dave Stewart here, who is the life sciences director of the UK synchrotron Diamond Light for X-ray crystallography. What he told us was that he'd heard from his friend Sehei Rao in China that the Chinese synchrotron was being maintained and was not operational. So we turned over the UK synchrotron to making proteins, and the proteins were made in the university and taken down so we could start studying the structure and sharing that information back to the Chinese. So that was the first thing that we started to do. And then the other thing that we decided to do was to support the vaccine from the Jenner. So Sarah Gilbert and her team had already swapped the spike bit of the virus, the code for that, into chimpanzee adenovirus vectors called ChAdOx vectors that she and Adrian Hill have been working on for many years. And so she was already very much, the team and the Jenner were already much underway by the time we had this meeting. And we had a lot of confidence that that was going to work because Sarah had been working on this Middle Eastern respiratory syndrome, trying to make a vaccine to this. So MERS is a really nasty condition. It was first identified in Jeddah in 2012, and it affects camels and camel handlers mainly. There have been 5 cases in the UK. It binds a different receptor from the SARS viruses. This is a camel that's infected, and so Sarah had developed a vaccine to this. One of the most strange things I had in the first year that I was at Nuffield Professor is I had to sign off the purchase of a herd of camels so that this trial could be done. But that was kind of successful. So we had a confidence, but we didn't have any money at the time, because the way in which the vaccines were organized nationally, internationally, was that in 2017, an organization called CEPI, which is a coalition for epidemic preparedness and innovation, had been set up. And CEPI was charged with deciding where the money would go for vaccines in the event of a pandemic. And so they made some decisions to fund 3 vaccines. And at this stage, Sarah and the Jenna team, along with Kath Green, who runs our little clinical biomanufacturing facility, which is a really unique thing that we're able to make these vaccines locally. And this is a real treasure. But we made the seed stock, but we needed to get, or we were about to make the seed stock, but we needed to get a large manufacturing contract. And so the decision was, despite the fact we didn't have funding from CEPI, the university underwrote the program to £1 million so that we could get this underway. And that was kind of critical for getting the timelines going, because we realized we needed to press on. I remember that Adrian and Sarah said that we were asked them how long it would take to do a trial, and I think they said that they could start something in July. Well, they vastly exceeded that expectation. So we continued then with these meetings, weekly, trying to just progressively do what we could. I think that we were we're anxious to kind of do this in a fairly sort of quiet and but diligent way. We were interested in how we could affect testing, the Oxford vaccine, controlling transmission, so on. And I'll give you some of those examples now. So the first thing we needed to do was to track the virus. So the government decided at an early stage, Boris, to centralise the track and trace process, and they set up these lighthouse laboratories. So they came round, the army came round and took all our machines. So we couldn't do anything to screen for viruses anyway. They'd all went to Milton Keynes, and they started generating, screening at a reasonable rate. But as I said before, the track and trace thing didn't work terribly well, because I think we just were not prepared for it. So this was done by the government. We were just supplying equipment. What we did decide was that we wanted to contribute to understanding the distribution of the infection across the UK. And so one of our professors, Sarah Walker, she set up a large trial monitoring the infection in households across the UK. And that was run with the Office of National Statistics. So Sarah ran that trial. And she continues to run it. It's a huge effort, but it's a thing that's reported on the news at night. And she was awarded the OBE for that last year, and so that was our main contribution to tracking the virus. And we also started collecting large numbers of samples, and we collected the largest set of samples in the UK. That then meant that we were able to develop new technologies. So one of the engineers span out a company which was able which is now screening individuals as they come in directly through Heathrow, and slightly less fortunately, we also came to sort of that national place for testing kits that the government were buying in very, very large numbers, and some of them just didn't work basically. A lot of them were good, and they were then taken forward, but that we became, because we had all these samples and we were making antigens, we then became sort of national place for testing. The thing that we decided to make the biggest difference was to measure the immune response. So you need to know who's been infected, but then you need to know who's had the infection. And so to do this, we set up a pipeline of making testing antibodies. And we did this in collaboration. I was very keen to do everything with the NHS, and I thought that was a way to have a sort of more dispersed model. So we had the NHS as our front end. We then took the samples in to the department. We bought some, we spent in about £2 million on some robots, and which that was with government support, and we had a commercial partner, and we started doing antibody testing to look at who was immune. And we did some surveys. We're looking at healthcare workers, and then we bolted this onto the national survey. So the data that we would generated across the country was being done by us. And so since March of last year, we've done about 250,000 of these tests, which then contribute to the data that you see on the news at night. And we're now just about to do a further a study where we're going to do 300,000 tests a month, looking for people who have been vaccinated and people who have developed further infections in the general population. And what's going to happen is they're going to send out these little tiny tubes, vacuum tubes, and they're going to ask people to take finger pricks and send them back. So you may get one of these, I guess, in the post, and that's the work that we're going to continue doing going forward. And this is the kind of data that we generated. So this looks at the antibody results from June through to January, looking at the number of people who've been infected. You can see it's risen to about 15% in the UK population. And there's a distribution. There's a distribution across the country. So you can see that in Southwest, very relatively few. Most people, the highest instance is in London, where about 20% of people have had the virus before the vaccination program. Okay, so the next thing we wanted to do was we wanted to sort of see if we could delay the infection. And so, and there was a lot of talk about why the Chinese were so good at this. So we, in March, Christoph Fraser in the department came up with the idea that it might be possible to control the infection with mobile apps. And I mean, this gave us all a big boost because it seemed such a brilliant idea. This was then published in Science. There was a flurry of activity because he contacted Matt Hancock and Bill Gates, and the UK government then talked to people overseas. The putting into practice a bit harder, but we had the first app that was trialed on the Isle of Wight was developed by the NHS. There were problems with that. And then a second app was rolled out in August, and that's the one that we've been using since September. And the estimate as of this month is that it's probably saved about 600,000 cases. And there have been around 25 million downloads. So this kind of held that we felt that this kind of technology in the future would be something that might be able to hold pandemics. The other thing we did was we wanted to trial new drugs. So we set up this, Peter Horby again, set up the recovery trial. This is one of the fastest piece of draftings of a trial ever done, because he and his co-director Martin Landry managed to draft this by the 10th of March. So that was before the WHO had even declared the pandemic. We had this trial underway, and it was embedded in the NHS, which really made it possible. And the first patient was recruited on the 19th. So the first thing they did was they showed that hydroxychloroquine, which you remember Trump was saying was the way to stop coronavirus. That was shown not to be effective for active disease. And then on the 16th of July, they reported that dexamethasone was effective at reducing mortality in people who were already on oxygen or ventilated. And that was just another huge advance. And they went on to show that various other drugs, the antiviral drugs, weren't of benefit, which was also important, particularly as they were being used quite widely. And we can now calculate that recovery has probably saved more than 500,000 lives. So a very big contribution, I think, from the university's research, and something we're obviously very proud about. There are a number of other trials we've been involved in, but recovery is now international and continuing. So the vaccine, Adrian's already talked to you, I think, about the vaccine, so I'm not going to spend a lot of time on it. But the trials the animal work had got underway quite quickly, but we had to wait for safety data. And so the trials team, which was led by Andy Pollard here, you've probably seen him talking about this, and Maheshi Ramasamy, who's also a fellow here at Magdalen, they helped to run the trials and started recruitment in March. Adrian Hill and Sarah and a very able young PI of ours called Sandy Douglas started putting together a program for how we would manufacture the vaccine. And that sort of laid the foundation for everything that's done in the UK. And that, but it was very clear soon that we needed a partner. And so John Bell and Gavin Screeton negotiated first with Merck and then with AstraZeneca, and then signed off this deal with AstraZeneca. And it was on the basis that during the pandemic, the cost, as you know, would be, the price would be the cost with no profit. And that's why our vaccine is £3 and the Pfizer-BioNTech's around about £30. And the idea was that it would be possible to distribute that. And the latest this week is that it's been approved for distribution by WHO, which means it can be distributed to all the lower income, middle income countries. And this is just a map of where there's been approval. These countries that don't have the formal approval don't have the mechanisms, but will now be available to receive the vaccine. And Adrian did a lot of work making this possible, particularly with the Serum Institute of India, which is probably the biggest manufacturer of vaccines. And he said that they're now supplying 100 million doses to COVAX this week. So that's where that's going. We also were pushed ahead with the structures. So we realized that vaccines are too slow. So we've got a program to try and develop drugs against the coronaviruses, to pan-coronavirus drugs that we could then pull out in future pandemic threats. And so that's an ongoing process, and one which we're beginning to think about how we could commercialize that. But we've got various lead targets, and so that's an ongoing part. So by the first national lockdown on the 23rd of March, we had the vaccine trial was almost underway. Recovery trial was already recruiting. The mobile app idea was in conception and development. The serology screening was underway, and that was going to become the part of the Office of National Statistics survey. And we started our sort of drug discovery early phase, and that's something to take forward in the future. By the second lockdown on the 31st of October, the vaccine trial was well underway in South Africa and Brazil and the UK. We had the dexamethasone report, the mobile app was launched, the ONS data was still coming in. This drug discovery was now open, sort of open access, called Moonshot, and we were studying the disease in the patients in detail. And then just around Christmas, we started, the government were already sequencing large numbers of viruses, mainly at the Sanger Institute, but we were doing a little bit here in Oxford. And as a result of that, we began to get a bit of a clearer picture of how the virus was evolving. And I just want to show you this cartoon here about how this has affected the structure of the top of the spike. So this is the receptor binding domain of the spike, and it binds to the ACE receptor up here. So this is the binding face where the virus comes in and binds. And this is the peptide sequence here. And what I'm going to show you is little snapshots from March through until January, showing little mutations coming up. And there are two. This is a mutation that defines the Kent variant, and this is one that defines the South African variant. So I'll just play the video for you. So you can see that as time goes by, this is looking at the whole data from across the UK collected. You can see these little variants pop up just by chance, the mutations. And what you'll see, we're now getting through to June, but by the time we get through to about October, you will see this variant starting to pop up. And that's the Kent variant that becomes the dominant. And you occasionally see this South African variant just popping up at random, but it hasn't become dominant in the UK population yet. So so here we go. Here's the Kent variant coming up and being selected for as it becomes, and it's now becoming more frequent and eventually becoming becoming dominant. You see, there it is. And so that's how the virus has evolved over the last year. And this is just some data that we've generated in the department. Okay, so now the N501Y variant has come up and is now the main one in the population. And if you look at the structure of the spike more conventionally, you can see this is where the Kent variant mutation is. These are antibodies that bind, that Gavin Screeton has generated from patients. And you can see these are neutralizing antibodies here in red. You can see that why we're tolerant of this, but you can see also why this variant here from South Africa, affects the neutralization a bit more. And you can look at that in a bit more detail. So there's that's that bit where where it sticks up. And these are antibodies that bind and neutralize. And you can see how variation in the structure here might affect the binding of these antibodies. So this is something that we're continuing to work on for the government to help to inform sort of policy decisions and selection of what would be the next variants. So by the 6th of January, the vaccine trial had reported its 70% protection. We think it's going to be much better for preventing severe disease, as I've said. We had approval by the MHRA on the 30th of December, and we were surveying the different variant vaccines. On the 14th of January, the recovery reported no benefit of convalescent plasma. So a lot of people being treated with plasma from patients who'd recovered, in particular in the US, that showed not to be a benefit. But on the 11th of February, that's just recently, we showed that a treatment to block IL-6, which is an inflammatory cytokine, was added to the effective death of mesosine, reducing the mortality by 50%. So in the space of a year, we've managed to go from no treatment to a treatment that reduces the mortality by 50%. And now, of course, we've managed to vaccinate 15 million people in the UK. And I think things are looking quite good. So Magdalen, about a month ago, where next? So mass vaccination, we're going to focus on neutralizing antibodies, but we need the most important thing in the next few weeks, severity of disease. And I think many of us are very confident that's going to be affected well. We need to be vigilant. We need to make variant vaccines. So these are already being manufactured for the autumn. So we'll have those ready. Vaccines are too slow. So we also need to make new drugs for next time. And we're going to focus in Oxford to create a centre for pandemic preparedness, which will allow us to do all of these things in much the same way as we've operated together over the last year. We want to put this all together in one institute, and it's all part of an international collaboration. And so I'm going to stop there. We're a bit late starting, but thank you very much.
Thank you very, very much. That was absolutely fascinating. Can you unshare your screen so we can back on screen? Brilliant, thanks. Thank you very much. I've had lots of questions coming in, and one of the themes is about what you might call vaccine competitiveness. Concerns about the difference in the efficacy of the Oxford vaccine compared with the Pfizer vaccine and some of the other vaccines. Why is there that difference in efficacy, and does it matter?
So what they're measuring is the ability of the virus to neutralize the vaccine to induce antibodies that causes neutralizing antibody titers to block the receptor. And they probably all produce different total amounts of antibodies. They probably produce the same spectrum, but different amounts. And so if you start with more, you're probably able to neutralize more, in a probabilistic way, you neutralize more. And so they do vary, but and that translates across variants. So if you have a variant which is more different, then you'll get more loss. But the important thing really is whether that is going. That's quite separate, as I said, from whether or not it prevents the severe disease, and you've got to ask yourself as well about how quickly you can get the vaccines out there. So if you can distribute a vaccine which can be operational within a 6-month window and vaccinate a lot of people and prevent a lot of disease, then you can manufacture a new variant vaccine quite quickly that will then substitute for it. Well, that's way better, Dinah, than having a vaccine which is slightly better that you can't distribute. And the key thing about the vaccine that we've got is that it's easy to distribute and to make, and it's cheap. And so that overrides all these other issues. But the other thing is, we're probably not going to eliminate this virus. It's going to be something that it's not going to be like smallpox, because we're going to have to live with it. So the idea that we're going to be able to create a vaccine that will prevent transmission completely is probably not feasible.
There's been quite a lot of questioning of the efficacy of the Oxford vaccine. So it was in the news today that some people in some European countries have been not turning up to their appointments if they've discovered they were going to get the Oxford vaccine. And why do you think there's been that reaction?
I don't know. I mean, it's one of the worst parts of this is the sort of terrible commercial and sort of quasi-political nature of this. I mean, you had President Macron saying that he didn't think that it was effective, at a point where he didn't have any vaccine supply. Well, it seemed to us that was just, a mechanism for demand control. I mean, the whole thing is pretty nuts, actually. I would definitely, obviously I would take I would actually take whatever vaccine was offered to me, because its supplies are limited, and it's very effective against transmission, and you need to just it's very effective. I don't understand it. It's beyond me. But there you go.
What about the delay in the second jab?
Oh, that's now clear that we were completely right about that. So it's good. It's probably better if you delay it. So that was one of the things that they said about our vaccine, wasn't it? Yeah, we shouldn't be the UK approach. Now they're absolutely vindicated on that, and the WHO have now recommended this week that it should be delayed. So it's not only that's an international policy now. Completely right. Do
you think
what's going to happen with all these things? What's going to happen in the next few weeks? The proof will be in what happens in the next few weeks. And already it looks like the case loads are going down. And I think
do you mean in the UK?
Yeah, in the UK.
Do you think you can already see?
It's the only experiment we've got in Europe.
Do you think you can see the effects of the vaccine happening in the UK?
Well, people seem to think that they're just beginning to in older people, and we're going to get this data very rapidly over the next few weeks. Mm-hmm. It'd be nice to say for sure. We're going to get data about the new variants in Brazil, I think. So that's what Andy Pollard told me this week, so there's a bit of an experiment going on, but I think we can be. I feel more optimistic than I feel very optimistic, actually, and I think the severity of disease thing that's the main thing. The vaccine from AstraZeneca may be much better at protecting against the severity of disease. That's the two things that I've described in my talk. Yeah, are kind of separate.
The T cell response.
Yeah, or whatever it is. It may be.
So do you think when people get their second jab, will they all have the same vaccine for the second jab that they had for the first? Should you always give two of the same, or can you mix and match?
No, well, possibly, there's kind of some ideas that it might be good to mix some of them. That you might be some might be much better at boosting than others. And one of the things that if you talk to Adrian, he will tell you that these sort of combinations, he's worked on these for many times. And so there is going to be some sort of trial here in the UK to try and work on this. And to see, but at the moment, the advice is you stick to what you had.
There was some quite worrying stuff that came out about the South African variant and effect on the efficacy of the vaccines. What does that for you? Does that worry you?
And not well. A little bit in the sense that the neutralizing antibody titers, as I said on that diagram on the model, it affects the place where the antibodies bind. And so if they can't bind as effectively, they can't neutralize as effectively. And so, if you've got a very big titer of antibodies, it probably doesn't matter much. If you've got a smaller titer, the level, then it matters a bit more. And so, not surprisingly, these vaccines do seem to produce lower titers. But if you look at the Oxford vaccine and there are other vaccines that are very similar, and you look at how at their ability to prevent disease, it looks very good. So
even with the South African variant.
Yeah, well, so far as we can. The South African and the Brazilian variants are very similar. They're parallel, a nice example of parallel evolution, where the virus is mutating in different places to get to the same point of being very good at infecting cells. And if you look at them, what is true for the Brazilian variant is probably true for the South African. Yeah.
There was an announcement this week about the new challenge trial.
Yeah, vaccine.
What's your view on that? Were people actually being deliberately infected and then vaccinated?
Yeah, so that's something on which there are two views. I mean, one view is that it may produce some very useful data about vaccines. The problem with it is that it looks at a very early stage in the disease. So it might be possible to use it to screen new vaccines. It might be possible to use it to screen for drugs. I think that in my own view is that the more important experiment is that which goes on in the general community, and we've got a big experiment going on already, and you can get a lot of data from that. But it may produce some useful data in specific circumstances.
I'm going to go on for a few extra minutes. Obviously, I know some people won't be able to stay, but since we started a bit late, there's a lot of really interesting questions here that I want to ask you. I'll ask you all of them, but there's some really interesting ones. Question about diverting resources. You've described this amazing moment at the beginning of last year when the whole of the Department of Medicine swung like a juggernaut into the path of the disease, from fighting it on so many different fronts with vaccines, with treatments, with data and so on. Yeah. So you must have had to divert resources from lots of other projects. Did that involve difficult choices?
Not really. I think it was amazing. The department's been amazing. So I think at the beginning, in fact, at the beginning, one of the conversations, I was looking at the notes, was a bit of a concern that we might project ourselves wrongly, as if we were enjoying some sort of research bonanza at the time when people were suffering in parts of the world. And so we were rather very cautious about it initially, but everybody was just very keen to try and do something to help. And so it was not difficult to get people motivated to do this. As I kind of explained to you, much of the work that we do is overseas anyway in Oxford. And so we have a very international perspective. And I think partly a lot of the stuff that we've done has just kind of set us up. We were kind of waiting for this moment slightly. And so, it was not difficult to get people engaged. Then, of course, since we've had the lockdown, then a lot of our researchers had to come to a halt anyway. And so we just diverted our resources to this. But
I think for a lot of people in the UK, discovering all of this going on in Oxford, it's been like suddenly finding that you had a Rolls Royce you didn't even know you had. That this extraordinary operation that was here under our noses, and that most people know about.
We knew we were the best medical school in the world. No, but we are the best medical school in the world. I think we are. There's some very special things about Oxford. One of the things is a very flat structure. We don't have a very hierarchical structure. So when we all, maybe quite a lot of the time we kind of, we're not all working together, but when we all work together, it's an amazing experience. That's been very uplifting for everybody to work together. And these meetings, everybody's piling in with ideas. it's partly a structural thing about the way Oxford's structured. It's very different from anywhere else in the country, I think.
How do you think the structure helps?
I think it enables people to work together without feeling that they're restricted in any way, and because the departments are very strong in Oxford, because the money basically flows to the departments, it means that we're able to deploy resources very quickly. So we could make the decision to support the vaccine, we can make rapid decisions to deploy things. There's no structure or other related hierarchy that we have to go through to make these. So it's very rapid decision. We were onto this before, way before, even the funders had got themselves together. So we were funding activity within the university before, we could get access money through the Wellcome Trust or government or anybody else. So, I mean, I think it's very, very quick.
A number of questions here about other types of research. Has Oxford been doing any research on vitamin D or omega-3s? And what about long COVID?
So there is a big piece of work now done by the Department of Psychiatry on long COVID. And they reckon that maybe 30% of people get long COVID. That's an argument why you might consider challenge studies to be questionable, right? You wouldn't want to give young kids long COVID syndrome. So there are, these are some of the things that come into play. Yes, there's a big piece of work on long COVID. The very poorly understood, because it affects all age range. The vitamin D question, vitamin D's come up, and actually I was told that Recovery looked at vitamin D as to whether or not, because Recovery was, the prestige of Recovery is they wanted cheap treatments that could be applied worldwide. Dexamethasone is super cheap, so that was really a key part of that. So vitamin D was something they looked at. I was told that the feeling was that vitamin D might have some effect, but that there's no convincing trial data basically that it does. But no doubt in lockdown we're all vitamin D deficient. So if you take some vitamin D, by the time you get sick, I don't think it will make any difference, a vitamin D. But in lockdown.
Question about vaccination. If you've already had COVID and you've got antibodies, why do you need to be vaccinated?
Well, possibly you don't, but it won't hurt you, and it's a booster.
And what about side effects? There's a question here about whether the Oxford vaccine has severe side effects, and is that being researched? Is there a concern about side effects?
I mean, that's the point of the trials, Dinah, and there are no significant side effects that have been detected in thousands of people that have been given the vaccine. And if anything, you normally expect some sort of side effects when you have a vaccine and you develop an immune response, but those are typically mild, and there've been no severe side effects. The only side effects, were with other vaccines where, those side effects again related to sort of just delivery of the virus. There's no significant side effects that have been detected.
Okay, question that I think lots of people want to know about. Looking forward, do you have any data milestones in your own mind with regard to the pace of releasing from lockdown, and in relation to the university functioning on a face-to-face basis? When do you think we might be back to something like normal?
The critical thing is the next few weeks, when we know that the vaccine ameliorates the severity of disease significantly against the new variants. In other words, we've uncoupled the neutralizing question from the pathology question. When we know that for sure, then we'll have certainty that we can plan to stop the lockdown as soon as all the vulnerable people have been immunized. And if that were true, it could be quite soon, right? And so
what does soon mean?
Well, it could be it could be well before summer, couldn't it? It could be, but we need that data, and then I imagine that in the autumn we'll have a new variant vaccine, and we'll be vulnerable people will be immunized against that.
The government's being much more cautious now in the things that it says about the pace of slowing down, releasing lockdown.
They got it wrong last year, didn't they?
Yeah, so there's a kind of reluctance to give people more false hope, I guess. Do you think they're being too cautious?
No. I think it's sensible now. I mean, they got it massively wrong and they got it wrong over, you know, this latest lockdown. It was against advice, and it cost us big time. The hospitals were really overrun with patients.
You mean in sort of early January?
Yeah, it was terrible, and you needed to go on the wards and see what.
Were you actually on the wards, Richard?
Yeah. And I've been around wearing P2. Wearing those masks is really tough. The nurses are on for sort of shifts of 2 hours because it's so difficult, and then off, and then on. It's very demanding stuff, and there's a lot of patients very unwell, and there's still a lot of patients still on the wards, because although the cases have gone down, there's a lag. So,
but does it feel less intense than, say, a month ago?
Yes, now it's less. Yeah. It's less. We know that because we can see the wards are beginning to clear out a bit.
Right.
Yeah.
The last thing I want to say is not a question, but it's a comment by Patrick Davey.
Yeah, Patrick
makes sums up what everyone feels having listened to you. He says, "What a totally brilliant, inspirational talk! Real deep enthusiasm. Wonderful. Many thanks. I'm sure I speak for the whole audience."
Okay, well, thank you very much.
He does too, and it's been fascinating to listen to you. Thank you so much, Richard, and please, all the best of luck to you and the rest of the department in carrying on this extraordinary.
Yeah, not just our department, Dina, the whole of the medical school, but we should all, and the whole university, in fact. So we should all be proud of our university.
Thank you all very, very much. Thank you for attending, everybody. It's been a fascinating evening, and I'm sorry about the glitch at the beginning. Please join us for our next webinar. Details will be released very soon. Thank you.
Thank you.